In the process of ovulation, ovary releases secondary oocyte. The secondary oocyte then enters the oviduct. The egg cell, surrounded by granulosa cells called corona radiata. Between the egg membrane and corona radiata, there's a jelly layer called zona pellucida of its glycoprotein coat. This stage called capacitation and must be removed in female reproductive tract. Sperm will diffuse into the egg cell. It then triggers the acrosomal reaction once the head of sperm contacts zona pellucida. Hydrolytic enzymes digest zona pellucida and enables the entire sperms enters the egg. Fusion of egg and sperm plasma membrane triggers and release calcium ion. This causes the cortical granules to fuse with plasma membrane. The enzyme from granule harden the zona pellucida. This resists the entry of other sperms. The combination of haploid sets of chromosomes from two parents, female and male, will form a zygote.
Cleavage is the repeated mitotic division of zygote to form a solid ball of cell called morula. Cleavage of human zygote occurs within the fallopian tube. The first cleavage takes place about 30 hours after fertilization. It divides zygote longitudinally into two blastomers. The second cleavage occurs within 40 hours after fertilization. It forms in 4 blastomers. The third cleavage takes place about 2.5 days after fertilization. During this early cleavage, the young embryo moves slowly down the fallopian tube towards the uterus. At the end of the third day, the embryo reaches the uterus. It looks like a mulberry called morula. A fluid-filled cavity called the blastocele formed within the morula becomes a hollow ball of cell called blastula.
At the end of cleavage, the embryo has more than 100 cells arranged around a central cavity and has reached uterus. At this stage, the embryo is called a blastocyst, the mammalian version of a blastula. Clustered at one end of the blastocyst cavity is a group of cells called the inner cell mat, which will develop into the embryo proper. It is cells of the inner cell mat that are the source of embryonic stem cell line.
Blastocyst possesses an inner cell mat, which subsequently forms the embryo. The outer layer of the blastocyst consists of cells collectively called trophoblasts. This layer surrounds the inner cell mat's high and a fluid-filled cavity known as the blastocele. The trophoblasts give rise to the placenta. A blastocyst exits the fallopian tube and leaves the zona pellucida. The property of endometrium will change to a brief window, which can allow blastocysts to implant.
Implantation of the embryo is initiated by the trophoblast, the outer epithelium of the blastocyst. Enzymes secreted by the trophoblast during implantation break down molecules of the endometrium, the lining of uterus, allowing invasion by the blastocyst. The trophoblast also extends finger-like projections that cause capillary in the endometrium to spill out blood that can be captured by trophoblast tissue. Around the time the embryo undergoes implantation, the inner cell mass of the blastocyst form a flat disc with an inner layer of cells, the epiblast, and outer layer, the hypoblast. As is true The human embryo developed almost entirely from epiblast cells. Following implantation, the trophoblast continued to expand into the endometrium and form new membrane appear. Although these extra-embryonic membranes arise from the embryo, they include specialized structure located outside the embryo. As implantation completed, gastrulation begins.
Of the formation of 3 germ layers. The cleavage produces blastocyst, which consists of trophoblast and inner cell mass. The inner cell mass rearranged into epiblast and hypoblast. Epiblast is the upper or inner layer, meanwhile the hypoblast is the lower or outer layer. During gastrulation, cells of epiblast migrate inward through the primitive streak at midline, or invagination occurs through the primitive streak. Primitive streak is analogous to blastopore. Some cells move laterally or sideways will form mesoderm. Meanwhile, the others that migrate downwards will form endoderm, or known as displaced hypoblast. The epiblast that remain on top of the surface is called as ectoderm.
Was he not a drop of fluid which gushed forth? In relation to this topic, the word fluid in this ayah is semen which ejaculated into the uterus of a woman. This explains the formation of zygote, where the nucleus of sperm and egg fuse together. This ayah explains that the nutfah or blastocyst then implanted in the endometrium. The blood clot then developed into a lump of meat, which will then develop into definitive human form. This process related to the organogenesis process.